Lynch syndrome is the most common colorectal cancer predisposition syndrome, conferring approximately 70 to 80% lifetime risk of developing colorectal cancer. We originally thought that Lynch syndrome was relatively uncommon. Now, however, as more and more people have their genome sequenced or having panel sequencing, for instance, patients who have colorectal cancer, and also other cancers, endometrial cancer, stomach cancer, ovarian cancer. We now think that Lynch syndrome is actually much more common, affecting approximately 1 out of every 280 Americans, and in Scandinavia, studies suggest about 1 out of every 250. Lynch syndrome is seen worldwide, so although there have not been precise estimates for Asia or Africa or South America, we anticipate that the levels will be similarly high. Lynch syndrome is caused by mutations in DNA mismatch repair genes. These are sort of like a spell checker. So you can imagine that if you're copying like a document in Microsoft Word and you had to do it by typing in the words, you'd make some errors. And then if you copied a copy or a copy of a copy of a copy, that you'd accumulate errors. The same thing happens during cell division when they replicate their genomes in mismatch repair deficient cancers. Consequently, because the mutation rates are very high and enriched in specific sequences, you develop recurrent mutations. For example, the TGF-beta type 2 receptor is mutated in approximately 60 to 80% of colorectal cancers that have mismatch repair deficiency. In addition, these type of tumors, because they're specifically enriched in insertion deletion mutations, for example, adding like an extra T or subtracting an A, which introduces completely new amino acid sequences, are the most immunogenic. This is the basis why in advanced malignancy, patients with Lynch syndrome respond, about 30 to 50% of them respond to checkpoint inhibitors. So our approach was to try to go earlier in the paradigm. Advanced tumors have very immunosuppressive microenvironments. They have cells that sort of basically kind of try to suppress the immune response against the tumor. If you go earlier to the point where you have sort of single cells or very few cancer cells, you're actually most likely to have a strong effect. And therefore, we adapted a tumor mutation vaccine approach using the fact that these mutations are recurrent, and we wanted to make sure that this is safe and efficacious. So we used mouse models. The interesting thing about our result was that suggesting that a vaccine that had few as 4 really recurrent mutations was able essentially to prevent cancer in mice with Lynch syndrome. And the simplicity of that is exciting because it means that we might be able to translate it. And we're currently hoping to do a phase 1b trial. So we've submitted a proposal to the National Cancer Institute to be able to have a trial in Lynch syndrome patients with the goals of demonstrating, one, that a vaccine is safe, which we believe it will, but is important really for all phase 1 studies, and second, that it induces the immune response in these patients. If that trial is successful, that would be followed up by a phase 2 kind of 3 trial with a clinical endpoint that's reduction in colorectal carcinomas in these patients. If one looks at the actually number of cancers that occur in both the developed and less developed world, there are overall sort of fewer cancers actually per age, when you adjust because different countries have populations that are sort of aging. But most of the benefit actually in the reduction in cancer is really from prevention. So actually the excitement in particular with the vaccines for human papillomavirus, hepatitis B, and hepatitis C has encouraged this idea for a cancer vaccine for non-viral cancers. So I would make the point that because they have high rates of immunogenic mutations, if it takes 100 steps to go right to be able to make an effective cancer vaccine, I would advocate that Lynch syndrome is the right place to start. If we're able to do this successfully, I think we can broaden it actually to other diseases. So there are recurrent mutations that occur in sporadic cancers. First, actually, let me say that in the sporadic form of the disease, about 4% of all solid tumors are mismatch repair deficient. So it's a substantial number of patients who are at risk for secondary malignancies that could benefit from this. But more broadly, there are other genes, for example, KRAS, BRAF, FLT3, DNMTA, et cetera, others that are of recurrent mutations, and this approach would actually hopefully be amenable to using a more broad cancer prevention vaccine. That wouldn't, by any means, I'm not saying this would eliminate cancer, but the point is that even because the burden of cancer is so large, that in the same way that like a pneumococcus vaccine, is able to reduce the rates of pneumonia in elderly individuals, even if we're able to reduce the rates of cancer by a few percent, that would be quite a substantial boon to public health. The potential benefits of immunoprevention are the economics. We've seen the cost of therapies for advanced cancers has sort of gotten quite substantial to the point where patients really in the United States have to decide, can you afford to keep your house? You have to mortgage your house to be able to have your spouse, for example, receive some of the state-of-the-art therapies. The idea of immunoprevention, we're currently funded through the National Cancer Institute, and we'd like to try to make this as open source as possible and to be able to make this a low-cost alternative so that it could be of general public benefit.